So I'm very happy today to present this work, which we recently published. It's about soil microbiome diagnostics for successful inoculation with biofertilizers. First of all, I would like to tell you why I'm so fascinated with the microbiome, because it connects us to everything else around us. And so, for example, the microbiome in the soil is very important for the microbiome of plants. The microbiome of the soil includes nitrogen-fixing bacteria, such as rhizobia, but also fungi like mycorrhiza, which form a symbiosis with plants. And then this microbiome also can move inside the plant and also can be transmitted to the seeds and also, of course, to the food that we eat. So we eat plants, and our microbiome in our gut is influenced by what we eat. And the same is true for animals. So their microbiome is also influenced by what they eat. And it all comes back to the soil in the form of manure. And in conventional agriculture, we use mineral fertilizers, and they come with several problems. One of the problems is that they are mined, like the name indicates, and so these deposits are expected to be depleted in the next 100 years. The second problem of nitrogen fertilizer in particular is that they need a lot of energy to be produced. So they need high temperature, high pressure, and so this is also very costly for the environment. Finally, excess use of fertilizers can lead to eutrophication of rivers and lakes, so algal bloom, and this causes diversity loss. And that's why we are looking for alternatives, and one of them are biofertilizers, which are agricultural inputs which contain microorganisms that can promote plant growth. And these include nitrogen-fixing organisms. For example, soybean in Switzerland is always inoculated because the symbiosis partner is not present naturally in Swiss soils. And here you can see nodules with the bacteria. In this project, we work with fungi, and more specifically, arbusculum mycorrhizal fungi, which form a symbiosis with 80% of plant species. And in this symbiosis, they can colonize the inside of the plant roots, and they can form this characteristic arbuscule, which gives them their name, which means little tree. And this is where the exchange of nutrients takes place. So the plant provides to the fungi carbohydrates and lipids, and the fungi in return provides minerals and water. And in this project, we are trying to predict inoculation success with arbusculum mycorrhizal fungi. This was a project funded by the Gebert-Rüff Stiftung and a collaboration with Steffi Lutz from Agroscope, Klaus Schleppi from University of Basel, and Marcel van der Heijden from Agroscope. And in this project, what we did is we work with maize. We inoculate the maize in the spring with fungi, and then we let the farmer manage the field in the usual way. And then at the end of the summer, we collect the maize, and then we measure the yield, and we measure how successful the inoculation was. We also collected the roots, and we quantified the microbiome in the root. In the spring, we also sampled the soil and measured classical soil parameters and the soil microbiome. And our motivation for this work is to develop a tool where the farmers would enter the soil properties which they measure on a regular basis, for example, soil texture and pH and nutrients. And then this tool would be able to generate, first of all, the microbiome which is expected to be present in this soil. And then based on this information, we would be able to predict which biofertilizer is most likely to succeed in that particular soil. And so what we did is we performed 54 field experiments over 3 years. Here you can see a map of north of Switzerland with Zurich here in the middle. And we worked with the fungi called Rhizoglomus irregulare. And here you can see one of our collaborators inoculating the field. So these are on-farm experiments, meaning we are collaborating with farmers, and they are managing their field, and we're just adding a little bit of fungi. Here is a slide which shows all the results from the 3 years. On the x-axis, you have all the fields, which are ranked from most successful to least successful. And what we found is in 1/4 of the fields, we have a positive mycorrhizal growth response. We also have some fields with a negative mycorrhizal growth response. So we measured soil physical chemical properties, for example, soil texture, water holding capacity, pH, nutrients, and also biological properties like microbial biomass and microbial respiration. And then here is a principal component analysis of all this data, and we are showing the chemical and physical properties as well as the fields. And the fields here are colored by this mycorrhizal growth response. So here you can see no pattern. Basically, there is no relationship between the soil properties and the mycorrhizal growth response. We also sequenced the fungal community in the spring. And here is an ordination showing the fungal community and the fields. Again, they are colored by MGR. And here you can see a very clear pattern. So we have the fields with high MGR, where the inoculation was successful, which are clustered together, which means they have a similar community composition. And then we have the fields with low MGR, which are clustered in a different part of the plot, which means they have a different community composition. So we put together all these variables in multiple models, and here I'm just showing one of them. And here, in this model, we found that the microbiome explains most of the variation in mycorrhizal growth response. So here we see, for example, these two OTUs here, so these two soil fungi are positive predictor of the microbiome. We also have some fungi which are negative predictor. And then we also have some soil properties which we found to be either negative or positively correlated with the mycorrhizal growth response. So finally, we also harvested the roots, and we sequenced the microbiome of the roots. Here we use slightly different molecular methods, which favors the Glomeromycota, which are the arbuscular mycorrhizal fungi. And here what we found is we found that we could detect our inoculum in these roots. So we have specific markers for inoculum, and they are more abundant they are more abundant as expected. And then what's interesting is that they are replacing the native mycorrhiza. So they are suppressing the native mycorrhiza. So we analyze separately the field with low MGR and the field with high MGR. And here you can see that the same is true. We have our inoculum, which is very present, and more abundant, and it's also suppressing the native AMF. But what's happening differently in the high MGR fields is that we have also pathogens which are less abundant. So what we found is that AMF inoculation protects plants against colonization by putative pathogens. So to conclude, we found that the soil microbiome is a very good predictor for inoculation success with arbuscular mycorrhizal fungi. And this study was recently published last year. But we still don't really have a good diagnostic tool, because now what we did was the classical sequencing of the community using a target gene, but this is still very costly. So what we are looking for in the future is to develop a diagnostic tool to quantify these microbiome indicators so that we can better predict inoculation success. And finally, I would like to thank all the people who helped in this study. Also, lots of students helped for the field experiments, and funding from Gebert Rüsch Stiftung and the collaborators. Thanks a lot.
You have 1 minute for a question.
It's one of my favorite studies of 2023. But something that drew my attention now. Essentially, you show that mycorrhiza introduce an extremely aggressive invasive species that destroys the native biodiversity and destroy the yield in 25% of the cases. Shall we use mycorrhiza?
Thank you, Alex, for this tricky question. Yes, it's true that we have this. I mean, it is the species Rhizoglomus regularis is very common native species here. Our particular isolate is very aggressive. It's true, it is replacing the native AMF. The question is, and we have not answered this question, is what's happening the next year? So this is definitely something we have to test. So we have not done this yet. But yes, for sure. I personally love this work, but I'm also on other parts of my research trying to work about how to improve the native microbiome. And I think this is a really more promising avenue. Yes. Yes?
Thank you very much. One thing that I was noticing is almost all, you looked mostly at the mycobiome.
Yes, yes. We also are sequencing the bacterial communities right now. So this is a project from a master student together with Klaus Schleppi. So we have this data now. So we are improving the model using the bacterial community. Mm-hmm.
I have maybe the last question.
Sure.
When you say that you are able to build biomarket for farmer, do you think that you always have to have something personalized on each farmer, or can you build something that works for everybody?
Yeah, it is very tricky because what we found and others have found too is that it's very dependent on the context, and so very different from a mineral fertilizer that you can just apply and you know it will work. So I think it's a bit tricky, and yes, I think it's a bit difficult to this personalized medicine for agriculture. I'm not sure it's going to happen. For humans, yes, but for agriculture, I'm not sure. Yeah.
It's still not possible, but maybe in the future.
Maybe, yes, yes.
Okay. Thanks a lot, Natasha.